Welcome to Unit 2, and in this unit we're going to be learning everything about Unit 2 for AP Biology. So yeah, please feel free to join, and I'll see you on the other side. Let's look at a visual comparison now that we have both here. So prokaryotes. Prokaryotes lack internal membrane organelles. Okay, so they don't have things like a nucleus or the endoplasmic reticulum or the Golgi complex. They don't have these internal membrane bound organelles. Okay, but what they do have are they may have these specialized internal regions where certain chemical or metabolic activity happens. Now, eukaryotes have those internal membranes and they have membrane-bound organelles, meaning that I have the organelle itself. This is the mitochondria. Okay, it has an inner layer and it also has an outer layer. For the chloroplast too, it has an inner layer and it also has an outer layer. So it has membrane-bound organelles. And then it also, because it has these membranes, if I have the eukaryotic cell here, because it has these different membranes, it creates these partitions that puts the cell into like specialized regions. Now, I know this picture might look a little misleading, but prokaryotes are generally much smaller, okay, and structurally very simple. If I look at the size of this prokaryote, okay, it will be more or less the size of the mitochondria and the chloroplasts if I compare the sizes that way. Now, this sequence, okay, if we look at a very, very simplified sequence, the ancestral cell took in the prokaryotic cell, and that was done with endocytosis. Okay. Now, the internalized cell was not digested. So when we take in something with endocytosis, we create this vacuole around it. Okay. And usually we would send a lysosome to go and digest it, but this time it stayed undigested. And then these cells then formed a very, very long-term relationship. I mean, we still have mitochondria living inside us today. And then over time, this internal cellular structure became an organelle. And in this case, it became the chloroplast. Okay. And chloroplasts are the organelles that plants have because they can make their own food using photosynthesis. So let's look at the first piece of evidence, the double membranes. Okay, so if I have a double membrane, it means I have an outer membrane and I have an inner membrane. So how did this come to be? So if you think about it, in the process of endosymbiosis, we have a prokaryotic cell that is being engulfed or eaten up by a bigger eukaryotic cell. So it would pinch in with the process of endocytosis, okay? And it will form a vesicle around that substance that it took into the cell to be digested. But this time it was not digested. Okay, and then over time, these two cells formed a long-term relationship. Okay, a symbiotic relationship. And the internalized cell then becomes an organelle. So that is why the evidence for double membranes are so strong, because the outer membrane comes from the ancestral cell that took in the prokaryotes, and then the prokaryote had a single biphospholipid bilayer, and that stayed its inner membrane. Now, the other big evidence card is that they have their own DNA. Okay, now mitochondria and chloroplasts contain their own DNA, and they have circular DNA like other prokaryotes. Okay, so prokaryotes have circular DNA, and chloroplasts and mitochondria also have this circular DNA. Now, the organelle here has its own DNA. This supports endosymbiosis because the organelle, okay, the organelle once had its own genetic information. And it still has that genetic information, but once, when it was still free living, it had its own circular DNA. Okay. Evidence card 3, ribosomes. Okay. Now, mitochondria and chloroplasts, they have their own ribosomes. Okay. And remember, ribosomes make proteins. Now, ribosomes, especially the ones that we find within prokaryotic cells, resemble that we find in the mitochondria and also inside the chloroplast. Okay, meaning that these ribosomes are similar in structure. Now, this supports the idea because the ribosomes are also found in prokaryotic cells. Okay, so let's look at the next evidence, and this is also the last piece of evidence, so division. Okay, mitochondria and chloroplasts divide within eukaryotic cells, and they do that independently. Okay, meaning it's not just when the bigger cell divides that they divide on their own. Okay, and this type of division they do, called budding, also resembles the division of prokaryotic cells. So they would just split themselves in two. Okay, and prokaryotic cells do the same type of division. So because it resembles the division of prokaryotic cells, this supports endosymbiosis because the organelle divides like a prokaryotic cell. Okay, let's do a quick evidence summary of everything that we've learned. So the evidence that supports it is that they have these double membranes. And what these double membranes mean is that they have an outer membrane, okay? And this outer membrane is a biphospholipid layer. And then they also have inner membranes that are a biphospholipid layer. So they have these double membranes. And this is true for both the mitochondria and the chloroplasts. Prokaryotic cells do not always have a double membrane. Now, yes, some do have, okay, but most only has a single phosphobilipid layer. Okay. The other piece of evidence is that they have their own circular DNA, and this is true for mitochondria, chloroplasts, and prokaryotic cells. Another important piece of evidence is that they have the same type of ribosomes. Okay, so the ribosomes we find in prokaryotic cells, we find within chloroplasts, And we also find within mitochondria, they have the same type of ribosomes. And then finally, they divide independently. So the mitochondria, chloroplasts, and prokaryotic cells, they all divide independently. And they divide using the same process of kind of splitting in half, and we call that budding. Okay. If you enjoyed today's lesson, please don't forget to like and subscribe, and come join us on the Skool.com community page where you'll find AP Bio blueprints. And if you take a look inside, you will find that Unit 1 is completely free. See you there.